Okay, so welcome back to this next video in which we are discussing heterotrimeric G proteins and adenylyl cyclase enzymes. Okay, so we've discussed now the 5 different families of G protein coupled receptor, and we've now discussed the basic structure of a heterotrimeric G protein. What we now want to discuss is the different types of alpha subunit, the different types of beta subunit, and the different types of gamma subunit. So we'll begin with the most difficult one, which is the alpha subunit. So basically, in the human genome, there are 16 different genes for alpha subunits, okay? And when I say the human genome, what I mean is counting one copy of each of the homologous chromosomes. So I'm not talking about homologous chromosomes, basically. When I say the human genome, I mean one copy of each of the chromosomes. So one copy of chromosome 1, one copy of chromosome 2, one copy of chromosome 3, all the way along to one copy of chromosome 22. Okay, and we'll ignore the sex chromosomes for now. Okay, so on those 22 chromosomes, you will have 16 different genes which code for alpha subunits. And all of these genes will have different sequences of organic bases and will therefore code for proteins with different sequences of amino acids. Okay, so you end up with many different types of alpha subunit. Now, you actually end up with 21 different types of alpha subunit. Now this seems like a bit of an anomaly, okay, because if we've only got 16 genes, how on earth do we end up with 21 different subunits? Well, basically this occurs because of splicing variants, basically. So let's just talk about the central dogma of biology. Okay, so let's say we have our gene here, so this is the DNA, okay, and I will colour in red here the coding strand of the DNA. So remember, when RNA polymerase uses the DNA to produce an mRNA piece, basically it only needs one of these strands of the DNA. Okay, and this strand which you use to synthesize the mRNA is called the coding strand. And you build a piece of mRNA that has a complementary sequence of organic bases to this coding strand. The other strand of the DNA, which is complementary to the coding strand, which I'll show here in turquoise, which does not code for mRNA, is then called the non-coding strand. Okay, so what will happen is you'll take your gene here, you will open it up temporarily, so you'll break the two strands apart, and then you will synthesize a piece of mRNA which is complementary to the coding strand. So let me show this here, and I'll show this one in blue. So you have produced a piece of mRNA that is complementary to the coding strand, okay, and it's here, let's say. So here is this piece of mRNA which is complementary to the coding strand. Now, this piece of mRNA does not get translated immediately, i.e. it doesn't go directly into the cytoplasm and find a ribosome and produce a protein. Before that occurs, there are all sorts of post-transcriptional modifications that are going to occur, and one of the main ones is splicing. Okay, so basically you don't want to use all of this mRNA to actually make your protein. There are portions that you want to use, and these are called the exons. So let's say I'll draw some exons here. So this is an exon, that's an exon, and here's another exon. So these are all exons, okay? And exons are the coding pieces of DNA. Now, there are then portions in between the exons which are not going to be used to actually make the protein, and these are called introns, okay? Now, basically, what's going to happen is a process called splicing, where you're going to cut the mRNA up, okay, and you will then stitch back together the exons, okay, so you cut out the introns and then you stitch back together the exons. Now, the final product that you get isn't necessarily always going to be the same, okay? So there are different ways that you can splice the mRNA. For instance, there could be some exons which it is not necessary to actually include, i.e. you can include them if you want, but they don't have to be included. In addition, there may be some exons which have differing levels to which they can be included, i.e. if we take, for instance, this exon, you might not have to put the entire thing in. You might be able to chop it in half and only put half in. Okay, so the final mature mRNA that you produce from the original primary transcript here is basically not always going to be necessarily the same, basically. And you can produce different mature mRNAs from certain genes. And these different mature mRNAs will therefore code for differing proteins. Okay? And these different proteins that you end up with at the end are known as the different splice variants of the proteins. Okay, so you end up with different splice variants. Okay, right. So this is how one gene can actually make different proteins, basically, by these different ways in which you can splice the mRNA that you're producing. Okay, so that's how 16 genes can go to 21 subunits. Right, so let's now discuss the genes for alpha subunits. Okay, so I'll just get another piece of paper. Right, okay, so the genes for alpha subunits of heterotrimeric G proteins are grouped into families, okay? So there are 16 genes and they're grouped into 4 families. So the first family, is called the GS family, okay? And this well, actually, I'm going to just replace that with G alpha S, because we're only discussing the alpha heterotrimeric G protein subunits at the moment. Okay, so this is the G alpha S family of alpha subunits. And this contains 2 genes. It contains the gene G alpha S, and it also contains the gene G alpha OLF. Okay, now G alpha S has multiple splice variants, okay? G alpha OLF has only one known splice variant, so it only produces one protein. Now, the next family of alpha subunits of heterotrimeric G proteins is called the G alpha I/0 family, and this contains 8 different genes. Okay, so it contains 3 alpha I genes, so G alpha I1, G alpha I2, G alpha I3. These are all members of this family. Okay, and they're all separate genes. Then we also have G alpha T1, which is the alpha subunit of the heterotrimeric G protein called transducin that is found in rod cells. Okay, so this is very, very important in phototransduction. So we've talked about, mentioned the Radisson Photoreceptor earlier. Basically, that is coupled to a heterotrimeric G protein, and when light hits the rhodopsin receptor, it causes the activation of that receptor, and it can then act on its heterotrimeric G protein, and the alpha subunit of that heterotrimeric G protein is G alpha T1, at least in rod transducin. So the heterotrimeric G protein which photoreceptors interact with is called transducin, and the form that you find in rod cells has this alpha subunit, G alpha T1. Okay, there is also a form for cone cells, so the alpha subunit of the heterotrimeric G protein transducin within cone cells is G alpha T2, and this is in the same family again. So this is cone transducin. Okay, of course you need the beta and the gamma subunit as well. This is just the alpha subunit of the rod and cone cell transducin. Okay, now, what other genes do you have? You also have G alpha O, okay, so the other one after which the entire family is named. And G alpha O has 2 important splice variants, and because the naming is quite nice and simple for the G alpha O splice variants, I will give it. Okay, so the 2 splice variants of the G alpha O gene are G alpha OA and G alpha OB. Okay, and if you're reading the literature, you will hear about these basically. Of the splice variants, they are well understood. Okay, compared to, for instance, the splice variants of G alpha S, these splice variants are far better understood. Okay, right. Then we've got G alpha, which ones are next? We need G alpha Z next, and then finally, the final member of this large family of alpha subunits is G alpha gust. So these are all different genes coding for alpha subunits. So we have these three G alpha I's, G alpha I1, G alpha I2, G alpha I3. And I want to stress those are not just different splice variants of one gene. Those are three separate genes, okay? So you have 8 genes in total in this family. Right, the next family is slightly simpler. Okay, so the next family is the G alpha Q/11 family of alpha subunits of heterotrimeric G proteins. And in this family, what you have is the G alpha Q gene, the G alpha 11 gene. Okay, so the 2 genes after which the entire family is named. Then you have the G alpha 14. Okay, and then finally it gets a little bit more complicated with the naming. Okay, so basically, the final one is either called G alpha 16 or it's called G alpha 15/16. Now, why is it called this? Well, basically, people doing work in mice found a alpha subunit of a heterotrimeric G protein which they called G alpha 15. People doing work in humans found an alpha subunit of a heterotrimeric G protein which they called G alpha 16, but they found that this G alpha 16 is basically the human equivalent of the G alpha 15 in mice. Okay, so really they should have called this G alpha 15 in humans. The problem is they'd already named it G alpha 16, and by the time people realized that it was the same thing as we've been looking at in mice for so long, the name had caught on. So now we've got G alpha 16 as the name for the protein in humans, and G alpha 15 as the name for the protein in mice. So some people are trying to do a movement towards this name G alpha 15/16, because after all this is the same protein. This is just the mouse version and this is the human version. So it would be nice if we had one name for it. But certainly don't call it G alpha 15 unless you're talking about it within a mouse. If you want, you can use G alpha 16, but as I say, it would be nice if you used G alpha 15/16 to try and repair up this mess. Okay, right. Then the final family of alpha subunits is the G alpha 12/13 family of alpha subunits of heterotrimeric G proteins. And this contains G alpha 12 and G alpha 13. Okay, so there we go. There are the 16 different genes for alpha subunits. So let's just make sure we've got 16. 1, 2. There are 8 in this family. That takes us up to 10. 11, 12, 13. This is 1, 14, 15, 16. So those are the 16 different genes that code for different alpha subunits. And as I say, some of them, for instance, G alpha O and G alpha S, have multiple splice variants, which is how you then get up to a greater number of alpha subunits. So basically the message is that there are a huge number of different alpha subunits that you can put into your heterotrimeric G protein. So if you're building a heterotrimeric G protein, there is a vast plethora of alpha subunits that you have to play with. Now, the ones that generally people have heard of are G alpha S. People have generally heard of G alpha S. G alpha Q is very famous. And generally people just call this G alpha I, but of course there are 3 different types of G alpha I. But those are the 3 main famous ones. Okay, generally people haven't heard of the other ones. Okay, so they do exist. They aren't as well understood as the other ones. There's much less literature on them, but they do exist. Okay, so these are all the alpha subunits. Okay, right. Now let's talk about the beta subunits, because this adds an extra layer of complexity to how to build a heterotrimeric G protein. So, basically, there are 5 genes for beta subunits. Okay, and again, there are actually 6 different beta subunits that are known at present when I'm making this video. Okay, so again, we've got multiple splice variants of certain genes, well, of one gene, which produces 2 separate splice variants, and therefore takes us from 5 genes to 6 subunits. Okay, my pen is just run out, so I will call it there for this video, and we'll continue this discussion in the next video.